My name is Chen. I will introduce the paper submitted to iMetaMed. The title is The Future of Cancer Therapy, Nanomaterials and Tumor Microenvironment. The tumor microenvironment represents a dynamic and complex ecosystem. The composed of tumor microenvironment include heterogeneous cancer cells, an intricate tumor stroma, including the basement membrane, extracellular matrix, vascular system, and other structures, diverse stromal cells, and soluble signacules, such as cytokines and hormones, which collectively foster a protumorigenic milieu. Consequently, the TME plays a pivotal role in the acquisition and maintenance of cancer hallmarks to varying degrees. Compared to strategies that directly target cancer cells, therapeutic approaches targeting the TME exhibit significant clinical advantages. Although strategies targeting tumor cells and those targeting various components in the TME share the ultimate goal of inhibiting tumor growth, prolonging patient survival, or achieving a cure, they differ significantly in their target sites, mechanisms of action, and induced side effects. Tumor targeting strategies are susceptible to the inherent genomic instability of tumor cells, thereby leading to the development of drug resistance during treatment. Components within the TME exhibit greater genetic stability compared to tumor cells. Modulating the TME can effectively ameliorate drug resistance developed by tumor cells during treatment, thus making TME regulation a more advantageous therapeutic target. Nanomedicine integrates nanotechnology with medicine, utilizing advanced nanomaterials and targeted drug delivery systems to achieve optimized region-specific biodistribution and therapeutic outcomes. Currently, more than 50 nanomedicine products worldwide have received regulatory approval for clinical use, and these formulations have significantly improved therapeutic outcomes in various diseases, including malignant tumors, infectious diseases, neurological disorders, and other clinical indications by leveraging the unique physicochemical properties of nanomaterials. Multiple aspects of the TME can be modulated to achieve enhanced tumor efficacy. As shown in Figure 1, nanomaterials primarily regulate the complex components of the TME through the following 4 key mechanisms to enhance cancer therapy, including metabolic interventions, targeting strategies, drug delivery nanosystems, and combination therapies. Current preclinical nanomaterial platforms designed for TME-specific modulation fall into 2 major classes: organic and inorganic nanomaterials, with each category presenting unique benefits and challenges in cancer therapy implementation. Despite extensive preclinical research on nanomaterials, the successful clinical translation of nanomedicines remains limited. The current challenges and difficulties include biosafety and long-term toxicity assessments of nanomaterials, uncertainties in vivo biotransformation and metabolic pathways, therapeutic efficacy variations caused by the spatiotemporal heterogeneity of the TME, obstacles in translating laboratory research to clinical practice, and insufficient selectivity in the precise regulation of TME components. In the extensive literature on nanomaterial-mediated TME modulation for anti-cancer therapy, researchers often overlook these issues. This has led to many nanomaterials remaining at the fundamental research stage, thus hindering further clinical application and patient benefit. Overcoming these existing obstacles and challenges will enable researchers to fully exploit the advantages of nanomaterials in precisely modulating the TME, thereby improving cancer treatment outcomes. To facilitate the translation of these technologies to clinical applications, researchers must continuously optimize nanomaterials and effectively address the key challenges associated with precisely modulating the TME. Summary. In this paper, we discusses the substantial impediments that the complexity and heterogeneity of the TME pose to the implementation of cancer treatment strategies, and systematically analyzes the principal factors contributing to the significant disparities in treatment efficacy and patient prognosis frequently observed between preclinical research and clinical trials of nanomaterial-based therapeutics. This paper underscores key challenges and difficulties currently faced by nanomaterials in cancer treatment, including issues related to nanomaterial biosafety and long-term toxicity assessment, uncertainties in vivo biotransformation and metabolic pathway, therapeutic efficacy variations caused by spatiotemporal heterogeneity of the TME, barriers from laboratory research to clinical translation. and insufficient selectivity in the precise modulation of TME components. Future progress requires interdisciplinary efforts to develop safer, smarter nanomaterials and robust clinical translation strategies to fully realize the potential of nanotechnology in cancer therapy.